Hi, I'm Yeun from the laboratory of Zhuohua Zhang at the Burnham Institute for Medical Research. Today, we will show you a procedure for how to detect protein ubiquitination. We use this procedure in our laboratory to study parkin, a ubiquitin E3 ligase, and its substrate. So, let's get started.
To begin this protocol, transfect cells with plasmids expressing both the target protein and an epitope-tagged version of ubiquitin. Let the cells grow for about 40 hours, then move to the laboratory bench to lyse the cells. At the bench, aspirate off the liquid medium and wash the cells twice with PBS. Next, lyse the cells by adding 100 microliters cell lysis buffer, which contains 2% SDS per 6 cm plate. Swirl each plate so that the buffer completely covers the cells. Next, use a cell scraper to collect the lysed cells and transfer them to a 1.5 milliliter Eppendorf tube.